I want you to put yourself in a very specific, a very familiar headspace for just a second.
Okay, I'm ready.
Imagine you are standing out on the edge of a freshly mown lawn. It's midsummer, the sun is just, beating down, and you take this massive, deep breath in.
Oh, yeah. You know exactly what that smells like.
Right. It is that incredibly sharp, deeply comforting, intensely green scent that just kind of hangs there in the humid air. I mean, for a lot of us, it smells like Saturday mornings.
Yeah, it smells like childhood, really.
Mm-hmm.
Or at least, a really well-kept neighborhood. It's practically a cultural touchstone at this point.
Exactly.
That scent is so universally recognized, and our association with it is almost entirely positive. I mean, it signals growth and warmth and just being outdoors.
Right. It immediately puts you at ease. But we actually need to completely unpack that feeling today. Because here is where the science gets incredibly interesting and honestly, a little dark.
Just a little bit.
Yeah, that comforting smell. It's not just some accidental byproduct of a lawnmower doing its job. When we look at the chemistry, and we have this massive stack of research in our dossier for today's deep dive, to prove this, you are actually inhaling the scent of sheer, unadulterated panic.
Yeah.
Chemically speaking, that smell is injury. You are breathing in a frantic, high-speed emergency broadcast system.
That is definitely a much more accurate way to look at it. I mean, to a plant, a lawnmower blade tearing through its tissue, or even just a caterpillar taking a single bite out of a leaf, it is a catastrophic, life-threatening event.
Right? It's not a haircut.
No, not at all. Cells are being physically ruptured. The structural integrity of the organism is literally failing. And so, in a matter of milliseconds, the plant begins venting this massive cloud of highly reactive, volatile molecules directly into the air.
It's crazy.
So what we call that fresh-cut grass smell is, in reality, a chemical siren just screaming that the walls have been breached.
It totally changes how you look at a simple chore like mowing the lawn. I mean, I feel guilty now.
You're running over a battlefield, basically.
Seriously. So our mission for today's deep dive is based on a fascinating array of sources detailing this airborne conversation among plants. And we are going to track the whole trajectory of this science.
Which is a wild ride.
It really is. We are going to explore the wild initial rise, the dramatic and pretty brutal academic fall, and then the highly rigorous scientific comeback of a concept that the media once enthusiastically labeled the talking trees hypothesis.
Right. And we have to be incredibly disciplined as we navigate this, because it is just so easy to fall into the trap of romanticizing nature.
Oh, for sure.
Our goal here is to strip away all those decades of media hype and anthropomorphism. We need to look at the undeniable mechanistic chemistry. We have to figure out what is actually happening when these botanical sirens go off.
Are they actually talking to each other?
Exactly. Are plants truly warning each other like good, altruistic neighbors looking out for the community? Or is the reality of the forest much more cutthroat?
Like they're just ruthlessly eavesdropping on each other's misery to save their own skin.
That is the central mystery we're tackling today.
So to really understand the tension here, we have to go back to the exact moment scientists first tried to prove that plants were actually smelling these airborne alarms. We need to set the scene in 1983.
A big year for chemical ecology.
Right, because if you were reading the newspaper back then, you would have thought we just discovered that the forests were full of whispering Ents from Lord of the Rings.
Yeah, 1983 was a massive watershed moment. It was the year that a really landmark paper was published in the journal Science by two researchers named Ian T. Baldwin and Jack C. Schultz.
And science is a big deal.
Oh, context is huge here. Publishing in Science is just about the highest peak a researcher can climb. It is a megaphone to the entire scientific world, and it basically guarantees that the mainstream press is going to pick up your findings.
So what was the actual experiment that got them into science? Like, what did Baldwin and Schultz do that was so revolutionary at the time?
Well, they designed what was a highly provocative experiment using poplar trees and sugar maple seedlings. The setup was essentially this. They took damaged seedlings, plants that had their leaves physically torn and mutilated to simulate an attack, and they placed them into sealed, airtight chambers.
Okay.
But they didn't put them in alone. They placed them alongside completely undamaged, healthy neighbors.
Wait, so they are just locked in a glass box together, like a terrarium?
More or less, yeah.
Yeah.
It was a completely closed system where they were sharing the exact same captive air supply. The roots weren't touching. There was absolutely no physical connection between the plants.
It's just the air.
Right. The only medium connecting them was the air. And what they found inside those chambers was completely astonishing.
What happened to the healthy plants?
The undamaged plants, which had, never been touched, suddenly shifted their internal leaf chemistry. They began rapidly ramping up their production of defensive compounds, specifically things like phenolics and tannins.
Okay, I know tannins from wine, but what do they actually do for a leaf?
Well, in a plant defense context, tannins are incredibly nasty for herbivores. When an insect eats a leaf rich in tannins, those compounds actually bind to the proteins in the insect's gut.
Oh, wow.
Yeah, and essentially ruins their digestion, meaning the insect gets zero nutritional value from the leaf and can actually starve to death with a full stomach.
That is brutal.
It makes the plant highly unpalatable. So Baldwin and Schultz are looking at these healthy plants, safely sitting in their own pots, suddenly pumping themselves full of toxins.
Just because they're in the same room?
Exactly. It was as if they had been chemically forewarned by the damaged plants sharing their airspace.
I mean, I can completely see why the press would lose their minds over this. You read the sources, and the Washington Post immediately ran a story featuring the phrase talking trees.
Of course they did.
It's such a beautifully romantic idea, isn't it? The idea that a forest isn't just a bunch of isolated columns of wood, but a living, breathing community. The trees are whispering to each other, looking out for each other, sounding the alarm when danger approaches.
The media frenzy was intense, and it perfectly captured the public's imagination because they tapped into a very deep, very human desire to see our own social structures reflected in nature.
We want them to be like us.
We do. But while the public was marveling at talking trees, the reaction within the actual scientific community was drastically, almost violently different.
Yeah, I have to admit, as much as I love the romantic idea, I'm looking at the methodology of the experiment itself, and I have to push back a little.
Let's hear it.
They put these plants in a sealed chamber. Trees don't grow in giant airtight Tupperware containers in the wild.
No, they do not.
Right. So if you are trying to prove how an ecosystem functions, how does locking plants in a stagnant, windless box prove anything about how they act in nature? Out in the real world, you have wind, you have rain, you have huge distances between trees.
That intuition you are having right now,
Yeah.
that is the exact same skepticism that triggered a massive backlash against Baldwin and Schultz. Oh, yeah. Your critique perfectly mirrors why the broader scientific establishment ultimately rejected the 1983 findings. Critics immediately zeroed in on the highly unnatural environment of those sealed chambers.
Because the air was trapped.
Exactly. In a tiny, enclosed box with zero airflow, volatile gases concentrate to levels that you would never, ever see in an open forest.
It's the equivalent of testing whether someone can hear a whisper, but instead of whispering across a field, you lock them in a phone booth and blast a foghorn.
That is a fantastic analogy.
Of course they are going to react to it. It doesn't mean they can hear a whisper in a field.
Exactly. The concentration of the signal was completely artificially inflated. And there were other significant issues with the paper, too.
Like what?
Well, critics cited incredibly small sample sizes, which made the statistical significance of their results highly debatable. When you only test a few plants, natural random variation can look like a pattern.
Right, you need a bigger group to prove it's a real trend.
But honestly, the methodological flaws weren't even the biggest problem. The most fatal error, philosophically speaking, was how the entire concept was being interpreted by the public and, arguably, by the tone of the research itself.
What do you mean by the philosophical error? I mean, we are talking about chemistry here, not philosophy.
In evolutionary biology, the two are deeply intertwined. The scientific establishment accused the entire talking trees narrative of reading human intention into what was almost certainly just a chemical accident.
Oh, I see.
Think about the phrase talking trees.
Yeah.
It implies that the injured plant wants to warn its neighbor. It implies an act of altruism.
And evolution doesn't really do altruism, does it? Unless it's your own offspring.
Exactly. Evolutionary biologists in the 1980s were deeply steeped in the framework of the selfish gene. They despise assuming altruism without absolutely ironclad proof.
Because natural selection is ruthless.
Ruthlessly driven by individual self-interest and survival. Think about the energy economics of a plant. Synthesizing these complex volatile chemicals takes a massive amount of metabolic energy. Why on earth would a plant spend its precious hard-earned energy to broadcast a warning to the tree next door? That tree next door is its direct competitor for sunlight, water, and soil nutrients.
Wow.
Warning your competitor makes zero evolutionary sense.
Okay, so if the damaged plant gets eaten anyway, and the neighbor uses the warning to survive and block out the sun, The damaged plant just aided in its own genetic replacement.
Precisely.
So the scientific community basically looked at this science paper and said, your math is weak, your experimental setup is completely unnatural, and your underlying logic is romanticized anti-Darwinian nonsense.
It was a brutal takedown.
Yeah.
And the professional fallout was severe. For roughly a decade after the 1983 paper, the entire topic of plant-to-plant airborne communication became radioactive.
A decade.
Yes. If you were a young, ambitious researcher looking to build a career, get tenure, or secure grant money, you absolutely did not study this. It was considered fringe science, it was largely unpublishable, and it was a surefire way to get laughed out of a conference.
That is so harsh.
We entered a literal scientific winter for the study of plant communication.
That is such a harsh reality of how science actually progresses. It's not just this straight line of discovery, you know? It's subject to trends and heavy peer pressure.
Absolutely.
So how do you possibly come back from a freeze-out like that? If an entire subfield of biology gets iced out because the claims were too romantic and lacked rigor, how do you force the scientific establishment to take it seriously again?
You have to completely change the narrative. You have to stop looking for altruistic talking between plants, which offended the evolutionary biologists, and start looking for something undeniable.
Something selfish.
Yes, you have to find a mechanism that is rooted purely in brutal individual survival and self-interest. You have to prove that these chemicals aren't a charity broadcast, but a selfish defense mechanism.
Survival of the fittest. Okay, so when does the thaw happen? When does someone figure out the selfish angle?
The paradigm shift really begins in the 1990s, spearheaded by the brilliant work of chemical ecologists like Ted C.J. Turlings and Marcel Dick. They looked at the mess of the plant-to-plant debate and decided to pivot. They realized that trying to prove plants talk to each other was just too noisy and controversial. So they decided to look at a completely different airborne relationship.
Which was?
They looked at the relationship between plants and insects. And in doing so, they uncovered a phenomenon known as indirect defense.
Indirect defense. How does that differ from direct defense? Like earlier we talked about tannins that ruin a bug's stomach. I assume that's direct.
Yes, direct defense is exactly that: toxins, thorns, sticky sap. The plant fights the herbivore directly. But Turlings and Dick discovered something much more complex.
What did they find?
They found that when a plant is attacked by a herbivore, say a caterpillar chewing on a leaf, it doesn't just passively leak a generic smell of damage. The plant actually actively synthesizes and emits highly specific, complex blends of volatile chemicals into the air.
Okay, actively making them.
And these chemicals aren't drifting aimlessly. They are perfectly chemically tailored to summon the natural enemies of whatever is currently eating the plant.
Right, let me make sure I'm wrapping my head around this.
It's a lot, I know.
If I'm a plant and I can't run away from the caterpillar eating me, and my own internal toxins aren't working fast enough, I literally broadcast a chemical scent that summons a mercenary to come kill the caterpillar for me.
That's the mechanism, yes.
It's like the plant is operating like a mob boss. Like, I'm taking damage over here, send in the cleaners.
Well, while we want to avoid humanizing them too much, that is a structurally perfect analogy for the mechanism. The plant is essentially broadcasting a targeted distress signal.
Wow.
It's putting a scent into the air that effectively says, hey, parasitic wasps, that specific species of caterpillar you love to lay your eggs inside, it is right here, chewing on my leaves. Follow the scent trail. Come get your dinner.
That is wild.
The wasps fly in, they locate the caterpillar, they inject their eggs into it, which is a pretty gruesome process for the caterpillar, by the way, and the plant is ultimately saved. This wasn't romantic talking. This was a highly targeted, ruthlessly self-serving bodyguard economy.
That is just incredible. And I can see why, because it's so obviously self-serving. The evolutionary biologists who hated the 1983 paper can't argue with it.
They couldn't.
It makes perfect, selfish, Darwinian sense for a plant to spend energy calling in a bodyguard if it saves its own life.
Precisely. It completely bypassed the altruism problem. But the true masterpiece of this era, the specific study that I think completely rehabilitated the field's credibility and set the gold standard for rigor, came a bit later, in 2005.
Okay, 2005.
This was a paper published in Nature, led by a researcher named Sergio Reisman, along with Turlings and several colleagues. And what made this study so mind-bending is that they took the bodyguard economy, which we thought was just an airborne phenomenon, and they took it completely underground.
Underground, you mean in the root system?
Yes.
But how do volatiles work in the dirt?
That was the absolute genius of it. They were studying maize agricultural corn plants. Now, corn is heavily preyed upon by a devastating agricultural pest called the western corn rootworm.
Rootworm, so it eats the roots?
Exactly. As the name implies, the larvae of this insect chew through the roots of the corn, and they do it completely hidden underground in the dense, dark soil.
Sounds hard to fight.
It is. What Rasman and his team hypothesized, and then rigorously proved, was that the moment the roots are damaged by these specific rootworm larvae, the corn plant actively releases a highly specific, volatile chemical signal directly into the surrounding soil.
But air is easy to move through. Soil is dense, it's wet, it's full of microbes. What is the chemical doing down there, and how did they even measure it?
Well, to your first question, the chemical actually diffuses through the tiny air and water pockets in the soil matrix, and its purpose is to recruit insect-killing nematodes.
Nematodes, those are the microscopic worms, right?
Yes, entomopathogenic nematodes to be exact. They are tiny worms that hunt down insect larvae, burrow inside them, and release bacteria that liquefy the insect from the inside out so the nematode can eat it.
Oh, that is disgusting.
It's absolute biological warfare. The corn plant releases a specific volatile, and we'll get into the actual chemistry of it in a bit, that acts as a beacon for these nematodes.
And how on earth do you prove that in a lab? You can't just put a glass box over a root system and watch the worms. They're underground.
They had to design incredibly clever underground olfactometers. Essentially, they built specialized root chambers with different arms branching off. One arm had rootworms eating corn, releasing the chemical. Another arm had completely undamaged corn.
Like a maze for the worm.
Exactly like a maze. They placed the nematodes in the center and just watched which way they traveled through the soil. Time and time again, the nematodes followed the chemical gradient specifically toward the damaged roots. They were following the plant's 911 call.
That is utterly fascinating. So why was this specific 2005 corn study the final turning point that brought the field back to life?
Because the science was rock solid, the methodology was flawless, and the results were infinitely repeatable across different labs.
You couldn't deny it.
Right. It proved, without a shadow of a doubt, that plants actively synthesize and deploy volatile chemicals for their own defense, even in the incredibly complex environment of the soil.
It wasn't just a leak.
No, it proved it wasn't just a passive leak of chemicals. It was a complex, highly evolved survival strategy. And once the scientific establishment finally accepted that plants use volatiles purposefully and selfishly, it paved the way to look back at the old talking trees idea with new credibility.
Like maybe Baldwin and Schultz were onto something, they just explained it wrong?
Exactly. Researchers had far better analytical tools now and a much more rigorous evolutionary framework to study it with.
Okay, so we've established that plants can hire mercenaries, and they definitely release intense, intentional chemical signals when damaged. But I am still struggling to visualize how a plant actually does this.
It's microscopic, so it's hard to picture.
Right. I really want to understand the language they are speaking. What actually are these chemicals, and how does a loaf, which to me just looks like a flat green solar panel, manufacture them on the fly?
To understand the language, we have to completely change our perspective.
Yeah.
We need to zoom all the way in. We have to shrink down and go inside the cellular structure of the leaf itself.
Okay, zooming in.
You really have to stop thinking of a leaf as a passive piece of green tissue. If you look at it molecularly, a leaf is a tiny, highly pressurized, incredibly sophisticated chemical factory.
Oh, a factory.
Yes. It is packed with assembly lines, separated compartments, and reactive raw materials.
Okay, I'm shrinking down. I'm inside the leaf factory. We know from the source dossier that this airborne conversation is primarily spoken in 4 main chemical dialects. What exactly makes these chemicals different from the normal stuff a plant makes?
The key distinguishing factor of all 4 dialects is volatility. All these compounds are built from the ordinary byproducts of plant metabolism, fats, hormones, carbon chains, but they've been specifically structured to be incredibly light.
So they float.
Exactly. Their molecular weights are low, and their structures discourage them from sticking together. This is what allows them to evaporate at normal, ambient temperatures and drift away on the wind. If they were heavy, they would just pool up inside the leaf.
Got it. They have to be light and airy to function as a broadcast. So let's walk through this chemical factory. What is dialect number 1? What is the first thing that happens when the factory gets attacked?
Dialect 1 represents the green leaf volatiles, commonly abbreviated as GLVs. This is the exact family of chemicals that produces your freshly cut grass smell from the intro.
Oh, full circle.
Exactly. I want you to think of GLVs as the raw, immediate smoke alarm of the plant world.
Okay, the smoke alarm. But the smoke alarm needs a trigger, right? How does a leaf actually know it's being damaged to set the alarm off? I mean, it doesn't have a nervous system to feel pain.
It doesn't know.
Okay.
It comes down to cellular architecture and the basic laws of physical chemistry. In a healthy, unbothered leaf cell, you have various membranes that keep everything highly organized.
Like internal walls.
Yes, and these membranes are constructed from a lipid bilayer, essentially, fatty acids. Now, tucked away in separate, isolated compartments within the cell are highly reactive enzymes. As long as the plant is healthy, the fats and the enzymes are strictly separated by physical walls.
So it's like keeping the match completely separate from the striking surface?
That's a great way to picture it. But when a caterpillar takes a bite or a lawnmower slices through the grass, that physical wall is violently breached. The structural integrity of the cell membrane collapses.
The fortress wall comes down, the compartments mix, and chaos ensues.
Utter chaos at the molecular level. The moment those structures rupture, the enzymes mix with the fatty acids of the destroyed membrane. Specifically, an enzyme called lipoxygenase goes to work.
Lipoxygenase?
Yes. This enzyme acts like a set of molecular shears. It attacks the long chains of fatty acids and starts aggressively chopping them up. This kicks off a frantic, microscopic assembly line known as the LOX pathway.
Chopping up the fat.
Right. Within seconds, literally seconds of the bite, it chops those long structural fats into tiny, incredibly light 6-carbon fragments.
And those 6-carbon fragments are the alarm. That's what we were smelling on Saturday morning.
Yes, that's it. The signature molecule that first puffs out is a 6-carbon aldehyde. That is the raw, pungent, sharp scent of cellular destruction.
Wow.
And the plant's internal factory is so sophisticated that even as the cell is dying, other enzymes can quickly convert this base molecule into slightly different forms, creating related alcohols and esters, which subtly change the scent profile.
But the key here is the speed, right? Because this reaction is purely driven by the physical breaking of the cell, it happens instantly. It acts as an immediate, generalized alarm to the whole area that violence is occurring.
Precisely. It doesn't require complex genetic transcription to start.
Huh?
The moment the teeth break the cell, the GLVs pour out. It essentially screams physical breach,
Yeah.
structural failure, something is eating us.
But because it is just the smell of broken cells, it is indiscriminate. A lawnmower, a deer, a caterpillar, they all trigger GLVs.
Exactly. It is fast and loud, but it isn't specific.
All right, so GLVs are the smoke alarm. They tell you there's a fire, but they don't tell you if it's a grease fire or an electrical fire.
Right.
But earlier, you said plants can summon specific wasps to kill specific caterpillars. A generic smoke alarm can't do that. How does the plant send a targeted message? What is dialect number 2?
If dialect 1 is the generic smoke alarm, dialect 2 is the highly targeted 911 call. This dialect is composed of molecules called terpenoids.
Terpenoids.
Yes. And to understand terpenoids, we have to look at how the plant actually identifies its attacker.
Yes, please explain this because it blew my mind in the notes. How does a plant, without eyes or a brain, know what is eating it?
It's all about chemical recognition. When a caterpillar chews on a leaf, it isn't just taking tissue away. It is also leaving something behind.
What is it leaving?
It's saliva. Insect spit contains specific proteins and compounds called elicitors. One famous example found in caterpillar spit is called volicidin. Yeah. The plant cells actually have specialized receptor proteins on their surfaces that perfectly match the shape of these elicitors.
Like a lock and a key.
Exactly like a lock and key. When the caterpillar spit hits the torn edge of the leaf, the elicitor binds to the plant's receptor. This triggers a massive, complex chemical cascade inside the surviving cells.
What happens?
Calcium ions flood in, signaling pathways light up, and it eventually reaches the plant's DNA, turning on specific defensive genes. The plant analyzes the chemical signature of the saliva, identifies the exact species of caterpillar, and then boots up the factory lines to make terpenoids.
And how are these terpenoids actually built?
Terpenoids are the largest and most diverse family of plant volatiles. They are constructed using a brilliant modular system. I want you to think of it like a biological printing press that only has one type of stamp.
Okay, one stamp.
Right, a 5-carbon block called an isoprene unit. The plant uses 2 distinct internal metabolic pathways to churn out these 5-carbon blocks.
Okay, so the factory is just stamping out these 5-carbon chunks over and over.
Right, and then depending on which genes were activated by the bug spit, the plant starts assembling these blocks. If enzymes stitch 2 of these 5-carbon units together, you get a 10-carbon molecule known as a monoterpene.
Okay, monoterpene.
A very common one is ocimene, which has a sweet, herbaceous scent. If the factory stitches 3 units together, you get a slightly heavier 15-carbon molecule called a sesquiterpene.
Wait, I recognize that term, sesquiterpene. We talked about Raslin's corn study earlier. Was the underground nematode signal one of these?
You have an excellent memory. Yes, it was. When the corn roots identified the rootworm saliva, they started assembling 3-block units to create a highly specific 15-carbon sesquiterpene.
And it was just heavy enough to stay in the dirt.
Exactly. That molecule is heavy enough to persist in the soil, but volatile enough to diffuse and attract the nematode.
So the plant uses this 5-carbon printing press to build a custom molecule on demand.
Exactly. And because the plant can string these blocks together in thousands of different configurations and then twist them into rings or add oxygen atoms, it can create highly specific volatile blends.
It's like a recipe.
It is. The specific mix of monoterpenes and sesquiterpenes drifting into the air acts as a customized, personalized 911 call that summons the exact species of parasitic wasp that preys on that exact species of caterpillar.
That is genuinely one of the most astonishing things I have ever heard. The plant literally analyzes the spit of the bug currently eating it, runs that data to its DNA, boots up a modular chemical printing press, and broadcasts a personalized bounty hunter call.
It is a marvel of evolutionary engineering, it really is. Now let's move to dialect 3, because the plant isn't just worried about insects. It also has to worry about invisible threats.
Like diseases.
Yes. Dialect 3 revolves around a molecule called methyl salicylate, or MESA for short.
Salicylate, that root word always jumps out at me. We are talking about salicylic acid, right? The active ingredient in aspirin.
That is the exact molecule. Salicylic acid is a fundamental compound in biology. In humans, we use it to lower fevers and reduce inflammation. In plants, it serves a remarkably similar role.
It cures their fever.
Well, it is a crucial internal hormone used for immune defense, specifically against pathogens like viruses, bacteria, or fungal infections. When a leaf detects a virus, it floods itself with salicylic acid to fight it off.
That makes sense.
But there is a logistical problem for the plant. Regular salicylic acid is a relatively heavy, sticky molecule. It likes to form hydrogen bonds with water.
Meaning it doesn't evaporate, it stays liquid and stuck inside the vascular system of the plant.
Precisely. It is strictly an internal chemical. So how does a plant warn the rest of its leaves that a virus is attacking if the hormone is too heavy to fly?
I don't know how.
The plant modifies it. It utilizes an enzyme to attach a tiny 1-carbon molecular tag, a methyl group, onto the salicylic acid.
What does adding 1 carbon do? Does it make it lighter?
In chemistry, structure is everything. By capping the molecule with a methyl group, it prevents the oxygen atom from forming those sticky hydrogen bonds with water.
Ah.
Suddenly, the molecule's boiling point drops dramatically. The plant essentially gives the heavy hormone wings. It becomes methyl salicylate, which is highly volatile and instantly evaporates into the air.
So the plant takes its internal medicine, puts a molecular jetpack on it, and vents it into the wind. It's like an aspirin-scented memo.
That is a brilliant way to phrase it, honestly.
Yeah.
A recent, incredibly detailed 2023 study published in Science mapped out exactly how this transaction works.
Oh, recently. What did they find?
The methyl salicylate floats through the air, traveling rapidly between the branches of the infected plant or even blowing over to neighboring plants. When this airborne memo lands on healthy, uninfected leaf tissue, it enters the cells. The healthy tissue then deploys an enzyme that chemically snips the methyl group back off.
It unpacks the memo. It turns it back into regular salicylic acid.
Exactly. It strips the jetpack off, reverting the molecule back into the active, heavy immune hormone. And once that hormone is active in the healthy leaf, it triggers a massive physiological shift called systemic acquired resistance, or SAR.
SAR. So it raises the shields.
It essentially puts the entire plant and potentially any neighbor that intercepted the memo into a broad, long-lasting state of high alert. It raises the biological shields before the virus ever reaches them.
Okay, this is coming together beautifully. So dialect 1 was GLVs, the instant structural smoke alarm. Dialect 2 was terpenoids, the modular 911 call to summon bodyguards. Dialect 3 is methyl salicylate, the airborne aspirin memo warning about disease.
Spot on.
Which brings us to the final dialect. What is dialect 4?
Dialect 4 operates on the exact same mechanical principle as the third, but for a completely different threat. It is called methyl jasmonate, or MEJA.
Okay, jasmonate.
If methyl salicylate is the airborne memo for disease and viruses, methyl jasmonate is the airborne memo for physical wounds and chewing herbivores.
Let me guess the chemistry here, based on the last one.
Go for it.
The plant has a heavy internal hormone used to fight insects. It needs to warn the rest of the plant, so it uses an enzyme to slap a methyl group onto it, stopping the hydrogen bonds, making it volatile so it can fly through the air.
You have completely grasped the biochemistry of methylation. You are absolutely correct.
Yes.
The base molecule is jasmonic acid, which is the absolute master regulator of a plant's anti-herbivore defense system. When jasmonic acid levels spike, the plant starts churning out those digestion-ruining tannins we talked about way earlier.
The ones that starve the bugs.
Right. By converting it into methyl jasmonate, the plant creates a powerful airborne distress signal that can travel far and wide. In fact, this specific chemical is the absolute darling of plant research labs around the world.
Why do researchers love this one so much?
Because it is the perfect, non-destructive experimental tool. If you're a scientist and you want to study how a plant's genome reacts to being eaten alive by a caterpillar, maintaining a colony of live caterpillars and standardizing exactly how much they chew is a logistical nightmare.
Yeah, bugs don't follow instructions.
Exactly.
Yeah.
But you don't actually need the bug. You can just waft some synthetic methyl jasmonate vapor over a healthy plant in a glass chamber.
And the plant just reacts.
The plant's receptors will detect the vapor, completely freak out, and trigger the entire massive cascade of defensive genes without a single actual bite being taken. It perfectly mimics the systemic alarm of a bug attack.
Okay, I want to pause and synthesize all of this because realizing that a leaf operates as a complex chemical factory is truly staggering.
It's a whole different world.
If I am a plant and a caterpillar bites me, my cellular walls break, instantly venting green leaf volatiles, the smoke alarm that screams structural damage.
Yep.
Then my receptors analyze the elicitors in the caterpillar's saliva, boot up the 5-carbon printing press, and pump out custom terpenoids, the targeted 911 call, to summon the exact parasitic wasp that eats that caterpillar. Finally, to ensure the rest of my own leaves are ready for the war, I methylate my internal defensive hormones, salicylic and jasmonic acid, giving them wings to fly through the canopy as company mammals, putting every other branch on high alert.
That is an exceptionally clear and accurate summary of the 4 dialects of the airborne conversation.
But wait, here is where we hit the absolute core debate of the sources in our dossier. We have spent this time establishing exactly what the chemicals are and the brilliant molecular mechanics of how the leaf makes them.
Right.
But we still have to confront why they are released into the open air, which brings us all the way back to the tension that destroyed the 1983 paper.
The altruism problem.
Yes. We know the plant is acting selfishly to call in bodyguards or warn its own branches, but what happens when the neighbor smells it? Are they communicating or are they eavesdropping?
This is the crux of modern chemical ecology. Having established the chemistry, we must rigidly adhere to the evolutionary definitions of these interactions.
Okay, hit me with the definitions.
In behavioral ecology, there is a very strict rule. A signal that evolved specifically to convey information, because it benefits the sender, is a true communication. But a chemical cue that is released for another reason and only benefits the receiver is just a byproduct. It is not communication.
Let me play devil's advocate for a second.
Please do.
If a plant is injured, it leaks all these volatiles, the smoke alarms, the jasmonate memos. If a neighboring plant smells that leakage, decodes it, and uses it to arm its own defenses, why can't we call that communication? Information was transferred, a reaction occurred.
Because you have to look at the fitness payoff. In the scenario you just described, the injured plant, the sender, gains absolutely nothing from the neighbor's reaction.
It doesn't get anything back?
Nothing. In fact, it might be harming itself by helping a competitor survive. The injured plant is still being eaten by the caterpillar. The only organism that benefits is the receiver, who gets a free early warning system. Evolution doesn't select for traits that cost energy to help competitors.
Okay, I see the distinction. So, to use an analogy, if my neighbor's house catches on fire, and I smell the smoke drifting over the fence, and I immediately run out and turn my garden hose on my own roof to protect my house from the sparks my neighbor didn't communicate with me.
That's right.
They didn't intentionally send the smoke over the fence to do me a favor. Their house is just unfortunately burning down. I simply eavesdropped on their absolute disaster, recognized the cue of smoke, and used that information to save myself.
That is a phenomenal analogy, and it perfectly encapsulates how modern science views plant-to-plant airborne signaling. Your neighbor's house fire is the plant being violently chewed by a caterpillar.
And the smoke is the GLVs.
The smoke drifting over the fence is the uncontrollable venting of green leaf volatiles and methyl jasmonate. You grabbing the hose is the neighboring plant detecting those chemicals and ramping up its own defensive enzymes. It is pure, opportunistic eavesdropping.
That completely flips the romantic talking trees narrative on its head. It's not a cooperative utopia. It's a paranoid neighborhood where everyone is spying on everyone else's misfortune.
Very well put.
But how do we actually know this for sure? The 1983 paper failed because they used airtight Tupperware boxes. How do you prove that plants are actually eavesdropping in the chaotic real world where wind scatters the smoke?
You have to do the absolute hardest, most tedious and punishing work in all of biology.
Yeah.
Long-term, rigorous field experiments in a harsh natural environment.
Out of the lab.
Way out of the lab.
Yeah.
And that brings us to the monumental work of Richard Karban. Over the course of more than a decade, starting around the year 2000, Karban and his colleagues, which interestingly included Ian Baldwin, who was attempting to rigorously redeem his 1983 work, set up an incredible series of field experiments.
Where did they go?
They didn't use a lab. They went out into the Great Basin Desert of Utah.
The Utah desert. That is about as far from a stagnant, sealed lab chamber as you can physically get. I mean, you have extreme heat, massive wind, and totally open air.
Exactly. It is a completely ecologically valid setting. They were studying two distinct species of plants that naturally grow in close proximity out there. Big sagebrush, formerly known as Artemisia tridentata, and wild tobacco, Nicotiana attenuata.
Sagebrush and tobacco.
Right. And the experimental design was brutal in its simplicity and tediousness. Karban would go out into the scorching desert heat, walk up to specific sagebrush plants, and painstakingly clip their leaves with a pair of scissors to mechanically simulate the tissue damage caused by a chewing insect.
So he is just standing in the middle of the desert, acting like a giant metal caterpillar, giving the sagebrush a haircut.
Pretty much, yeah. He would clip them to force the release of those volatile damage signals. And then he carefully monitored the wild tobacco plants growing nearby plants that shared nothing but the open, windy desert air with the clipped sagebrush.
Okay, and what happened? Did the tobacco smell the scissors?
What he documented in his landmark papers in 2000 and 2006 was an absolute revelation that forever changed the field. They absolutely did smell it. Over the course of the season, Karban found that the wild tobacco plants growing immediately downwind of the mechanically clipped sagebrush suffered significantly less damage from natural insects compared to tobacco plants growing near healthy, unclipped sagebrush.
So it worked, even in the wind.
It worked. The tobacco plants were definitively detecting the volatile distress cues bleeding from the wounded sagebrush and reacting to protect themselves. But here was the most crucial, elegant detail of the entire study.
What's that?
The tobacco plants didn't just panic and flood their leaves with toxic defenses immediately upon smelling the damage.
Wait, why not? If you smell the neighbor's house on fire, why wouldn't you turn on the hose right away?
Because, as we discussed with the evolutionary constraints, manufacturing defensive chemicals like nicotine, in the case of tobacco, is incredibly metabolically expensive.
You're right, the energy cost.
It drains massive amounts of nitrogen and carbon that the plant desperately needs to grow roots or produce seeds. If a plant overreacts to every faint smell of damage and produces toxins unnecessarily, it will stunt its own growth and get outcompeted.
So what did they do instead?
Instead of fully arming themselves and wasting energy, the tobacco plants did something much smarter and much more subtle. They became primed.
Primed, like priming a pump before you turn it on.
Precisely. At a molecular level, priming is fascinating. The plant receives the airborne warning, but it doesn't pull the trigger. Instead, it gets its internal cellular machinery ready.
Oh, so?
It might undergo epigenetic changes, slightly unspooling its DNA where the defense genes are located so they can be read faster. It gathers the raw materials. It puts its immune system on a hair trigger.
So it's less like turning on the hose and flooding the yard and more like standing by the spigot with your hand firmly on the valve, muscles tensed, just waiting to see a single spark land on your roof.
That is exactly what priming is. And the result is that if an insect did eventually land on that primed tobacco plant and take a single bite, the plant's chemical response was massively faster, and the resulting toxicity was much higher than a plant that hadn't been primed by the neighbor's warning.
It saves energy until the absolute last second.
The primed plant deploys its weapons in minutes instead of hours.
That is an incredibly sophisticated biological strategy. It balances the need for defense with the harsh economics of survival.
It is brilliant. And crucially, this is where Karban's linguistic framing finally rescued the entire field of study from the ghost of 1983. In his papers, he explicitly refused to use the term talking trees.
He killed the metaphor.
He had to. He framed this entire complex interaction strictly as eavesdropping. The sagebrush was bleeding volatiles simply because it was hurt. It had no intention of warning anyone. The wild tobacco had simply evolved a highly sensitive molecular nose to detect that specific ambient scent of damage, allowing it to selfishly prime its own defenses.
It's a completely selfish mechanism. The tobacco is exploiting the sagebrush's pain.
Exactly. And that framing completely satisfied the evolutionary biologists. It proved the airborne phenomenon was absolutely real in the wild, while rigidly adhering to the ruthless, non-altruistic rules of natural selection. It gave the field the intellectual foundation it needed to explode into the massive discipline it is today.
This brings up a really massive debate that runs through our entire source dossier, and it's something I want us to tackle head-on.
Okay, let's get into it.
It's this whole idea of plant intelligence.
Yeah.
You look at the science we just discussed, plants analyzing insect saliva,
Okay.
deploying modular chemical printing presses to summon specific bodyguards, eavesdropping on their neighbors, and epigenetically priming their DNA based on olfactory memory.
That sounds smart.
It sounds brilliant.
Hmm.
Because of these discoveries, you see books and articles all the time now arguing that plants are intelligent, that they communicate, that they have memory. Why are mainstream scientists so deeply resistant to these labels? If a plant can do all that, why is the word intelligence so risky?
It is a fiercely debated topic, and it all comes down to the danger of anthropomorphism, our deeply ingrained human tendency to project our own traits, motivations, and cognitive experiences onto non-human entities.
So we make them too human.
Yes. When a popular science writer calls a plant's survival tactics intelligent, or when they say plants communicate with intent, they are actively smuggling deeply human concepts into a biological process. They are smuggling in the ideas of intention, consciousness, choice, and altruism.
Let me push back on that, though. Are we just being arrogant? Are we just defining intelligence so narrowly that only things with a human-like brain can qualify?
That is the counter-argument, yes.
I mean, if a plant takes in external information, the smell of a wounded neighbor processes that information and alters its future behavior to ensure its survival, isn't that a form of intelligence? Why do we demand a nervous system for a behavior to be considered smart?
That is the exact argument made by the proponents of plant neurobiology, which is a highly controversial subfield. They argue that we're being too animal-centric. But the mainstream scientific consensus pushes back on that for a very specific reason: clarity of mechanism.
What do you mean by clarity of mechanism?
When you use a word like intelligence, the average person immediately assumes there's a thinker behind the thought. They assume the plant is making a conscious choice.
Right. We want to believe the forest is this magical, cooperative utopia where everyone is making decisions to look out for everyone else. We want the ents.
Exactly. It's a beautiful metaphor, and it sells a lot of books, but it is deeply, profoundly misleading to the public about how evolution actually works. Plants are undeniably marvels of chemical engineering. They possess biological mechanisms that are breathtaking in their complexity.
The 5-carbon printing press.
Right. As we've discussed, they can chemically analyze insect saliva. They can sense gravity, they can track the sun, they can adjust their root growth based on soil gradients. But they do not have nervous systems. They do not have centralized processing. They do not have intent.
So they are essentially vast, incredibly complex biological algorithms. If X happens, execute Y protocol.
Yes. To call an opportunistic, algorithmically driven eavesdropping mechanism intelligent communication obscures the true, brutal beauty of how natural selection actually forged these abilities over millions of years of blind trial and error.
Survival of the ones who didn't die.
Right. The plants that didn't automatically react to the smell of damage died. The ones that did survived. There was no conscious thought involved. Scientists resist the intelligence label because it replaces rigorous biological understanding with a comforting fairy tale. It makes the public think plants are just slow animals, when in reality they're something completely different and arguably much stranger.
That is a really powerful way to frame it. The truth is actually cooler than the fairy tale.
I think so.
So, zooming out from the philosophical debate, what does this all mean for the big picture of ecology? We've proven that eavesdropping definitively happens in the Utah desert with sagebrush and tobacco. The chemistry is real. Does this mean that every single forest on Earth is a giant whispering network of chemical alarms?
This brings us to the final frontier of the current scientific debate, and it is what ecologists call the scale caveat. We have to very carefully separate what we call lab truth from forest truth.
Lab truth versus forest truth. Okay, lay out the physics of that for me.
Let's start with the lab truth. In a laboratory setting, or even in a controlled agricultural field, the physical conditions are highly constrained. In a lab, you have a sealed chamber. The physical gap between the plant emitting the chemical and the plant receiving it might be 10 centimeters. The air is perfectly artificially still.
Like the 1983 boxes.
Exactly. In that specific environment, the volatile signals are incredibly dense, they don't disperse, and their effect on the neighboring plant is undeniable and massively powerful.
Right, the signal is trapped.
Now, take that exact same chemical signal and step into the physical reality of a wild, open, old-growth forest. Volatiles are, by their very chemical definition, highly evaporative gases. The absolute moment they leave the stomata of the leaf, they begin to disperse into the incredibly massive volume of the Earth's atmosphere.
It's the inverse square law of physics, right? As a gas expands outward from a point source in 3 dimensions, its concentration drops exponentially with distance.
Yes, the dilution is rapid and extreme. But dilution isn't even the biggest problem. A forest is a highly dynamic physical environment. It is windy. A strong gust of wind can literally blow the chemical distress signal in the completely wrong direction, meaning the plant directly next door smells absolutely nothing.
Just bad luck.
And furthermore, the atmosphere itself is chemically hostile to these specific molecules.
Hostile how? Are the chemicals breaking down?
Rapidly. Think back to those terpenoids, those 5-carbon blocks. They often contain double bonds, which make them highly reactive. Sunlight, specifically ultraviolet radiation piercing through the canopy and ambient atmospheric ozone, actively attack those bonds.
They break them apart.
They literally tear the volatile molecules apart. Depending on the weather, a highly specific chemical 911 call might be degraded into meaningless chemical noise within a matter of minutes or even seconds after leaving the leaf.
So the signal is simultaneously fading from expansion, scattering wildly in the wind, and being actively shredded by solar radiation and ozone. The odds of a coherent message reaching a neighbor a few feet away seem almost impossible.
This is exactly why we reliably see these plant-to-plant eavesdropping effects in very specific constrained environments, but struggle to prove them in wild forests. It worked for Karban in the desert with sagebrush, because those specific plants naturally grow closely packed together in dense patches, and the volatile he was tracking happened to be exceptionally stable.
So it's an exception, not the rule.
Right. We also see this phenomenon constantly in modern agriculture.
Well, sure, agriculture makes sense. A cornfield is basically a giant outdoor laboratory.
Exactly. If you plant a massive commercial monoculture of maize or lima beans or cabbages, you have millions of physically identical plants packed tightly together in rows, sharing the exact same genetic code and the exact same receptor proteins.
Oh, that makes it so much easier for them.
It does. If one cabbage gets bitten by a moth, the chemical signal only has to hop a few inches to hit an identical neighbor perfectly tuned to receive it. The signal-to-noise ratio is incredibly high.
But a wild forest isn't a cabbage field.
No, it is not. A wild, old-growth forest is beautifully chaotic. It has huge physical distances between trees. More importantly, it has hundreds of different species of plants, shrubs, and fungi, all constantly emitting their own baseline background smells, creating a massive wall of chemical noise.
Just a cacophony of scents.
Trying to smell a specific distress signal in a forest is like trying to hear a specific whisper in the middle of a packed football stadium during a touchdown. So while we know absolutely that the airborne channel is real and functions chemically, the idea that an entire diverse forest is meaningfully communicating over long distances via the air is highly debated by ecologists. It remains the least settled, most deeply argued part of the science today.
That is such an important takeaway for anyone trying to understand biology, or really any science. We have to appreciate the absolute magic of the microscale, the incredible leaf factory, the chemical printing presses, the 911 calls to parasitic wasps.
It's all real.
We have to marvel at the fact that a plant can analyze bug spit, but we have to do that without automatically exaggerating it to a macro scale that the physics of the real world just won't support.
That is exactly right. True scientific discipline means accepting the physical limits of a phenomenon. Admitting that the signal gets blown away by the wind doesn't make the cellular chemistry any less wonderful. It just makes our understanding of the ecosystem more accurate.
Okay, as we wrap up this deep dive, I want to briefly recap this incredible journey we've been on. We started in the 1980s with the incredibly romantic media myths of the talking trees, an idea that the public fell in love with but that the scientific community tore apart because it lacked rigor and improperly assumed evolutionary altruism.
In a dark time for the field.
But then, rather than giving up, researchers pivoted. Through the brilliant work on indirect defense discovering the bodyguard economy, where plants summon wasps, and through Karban's meticulous, exhausting field experiments in the Utah desert, the field came roaring back to life with unassailable proof.
Proof of eavesdropping.
Right. Plants aren't talking in the human sense, but they are constantly reacting. They are recruiting mercenaries, and they are absolutely selfishly eavesdropping on the chemical screams of their neighbors to prime their own defenses. It is a rigorously proven, cutthroat chemical ecology that is infinitely more complex than we ever imagined.
It really is a remarkable story. It perfectly illustrates how the scientific method works. It self-corrects, it discards the romantic flaws, and it eventually finds a deeper, stranger, and much more mechanistic truth.
But before we go, I want to leave the listener with something to chew on. There is one final mind-bending hypothesis buried deep in the conclusion of our source dossier. And I want to throw this out there because it completely reframes literally everything we've talked about today.
Oh, this is good. This is, without a doubt, perhaps the most fascinating open question in the entire field of chemical ecology today. It's the concept of within-plant signaling.
Okay, lay this out for me. What is within plant signaling, and how does it change the eavesdropping debate?
We've spent this entire deep dive talking about volatiles drifting from one plant to another plant, from the damaged sagebrush to the healthy tobacco. But I want you to think about the physical architecture of a single large organism, like a massive oak tree.
Okay, big tree.
It has an internal vascular system, the xylem and phloem, that carries water, nutrients, and chemical signals up and down the trunk and through the branches.
Right, the internal plumbing of the tree.
Exactly. But here's the problem, that internal plumbing is surprisingly, dangerously slow. The sap moves at a crawl compared to the speed of an insect. If a swarm of caterpillars starts aggressively chewing on a leaf on a lower branch, the leaf needs to warn the rest of the tree.
So it sends a signal down the stem.
But sending a heavy chemical warning through the internal sap, fighting gravity all the way up the trunk to the top branches, can take a very long time.
Too long. The caterpillars will have eaten half the lower canopy by the time the top of the tree even knows they are there.
Exactly. The physical constraint of the vascular system is a massive vulnerability. So evolutionary biologists looked at this and asked a brilliant question. What if the true original evolutionary purpose of all these airborne chemicals isn't about warning neighbors at all? And what if it isn't even about neighbors eavesdropping? What if venting these highly volatile chemicals into the outside air is simply the fastest, most efficient way for a large plant to communicate with itself?
Oh, wow. I see where this is going.
Air moves exponentially faster than sap. If that lower branch gets bitten, and it instantly vents a massive cloud of green leaf volatiles and methyl jasmonate into the air, the ambient wind can carry that chemical signal up into the upper canopy of the exact same tree in a matter of seconds.
Because it just floats up.
Yes, the plant's own upper leaves have receptors to detect the airborne signal, and they instantly arm their defenses before the insect can even begin to climb up the trunk.
It's using the atmosphere as an external nervous system.
Precisely. It is outsourcing its internal communications to the wind.
So if that is true, when a neighboring plant detects the signal and primes its own defenses.
The neighbor isn't listening to a public broadcast. The neighbor might just be accidentally standing in the cross breeze of a plant frantically trying to talk to itself.
That is incredible. That is the ultimate selfish act. The plant is just trying to use the wind as a high-speed shortcut to warn its own branches, and the rest of the forest is just quietly listening in on a private intercom system.
It is an incredibly elegant hypothesis because it perfectly aligns with the strictest rules of evolutionary theory. The sender benefits immensely because it protects its own canopy and ensures its own survival. The receiver benefits by eavesdropping on the shortcut.
It perfectly solves the altruism problem.
It does. The entire mystery of why plants release these chemicals is solved without any need for altruism, community spirit, or human-like intelligence. It is just brilliant, ruthless biological engineering.
I love that so much. It makes the world seem so much more complex and alien. So to wrap this up, the next time you step outside on a beautiful summer weekend and you hear the distant roar of a lawnmower kicking up and you take a deep, relaxing breath of that sweet, comforting smell of freshly cut grass.
You'll never see it the same way again.
Exactly. I want you to remember what is actually happening at a molecular level. You aren't just smelling the pleasant essence of summer. You are standing right in the middle of a microscopic, desperate crossfire. You are inhaling a frantic, high-speed chemical conversation between a torn leaf and its own stem.